Regulatory T cells, commonly referred to as T-regs, play a critical role in maintaining order in the human immune system. These unique immune cells play an especially important role in the inflammatory response because they regulate and suppress excessive inflammation. Given the therapeutic potential of T-regs for patients with autoimmune disease, cancer, and organ transplants, both researchers and industry leaders are developing novel therapies that utilize the multifunctional efficacy of T-reg cells in disease treatments and improving health outcomes for patients. In the following presentation, Dr. Qichi Tong, professor and director at the Transplantation Research Lab in the Department of Surgery at the University of California, San Francisco, a scientific leader in T-reg function and clinical application, and Dr. Randall Alfano, vice president at Invitria, discuss the latest developments in T-reg research and clinical application. They explore the challenges and potential for T-reg therapy. In our first presentation, Dr. Tong will share the latest research conducted by her lab at UCSF using T-reg cells for therapy. Then, Dr. Alfano will discuss T-reg expansion in chemically defined media without using serum or blood-derived components, and why that is critical for future clinical development and success of T-reg therapy. A copy of each presentation will be available for download on the Invitria website.
I'm Qichi Tong, and I go by Tong. So I've been at UCSF since 2000, and about 10 years ago, Jeff Bluestone and we decided to start this T-reg cell therapy program here at UCSF. And so this talk, I'm going to give you some idea of our vision for this program and also our experience so far. So why do we work on regulatory T cells? And then it's because if you don't have these cells, and actually it's a human disease, and these young boys actually can have fatal autoimmune diseases. And then that tells us regulatory T cells is critical for immune homeostasis.
In the space of T-reg, essentially when these cells begin to lose expression of CD45RA, they actually have a really bad habit of also downregulating FOXP3, which is caused by repetitive rounds of stimulation ex vivo. We actually have a starting medium formulation known as OptiPeak T lymphocyte, totally chemically defined, and it's optimized for the expansion of normal conventional T cells in humans.